Hi everyone, welcome to your unit on Mendelian genetics. Genetics is the study of inheritance. I want you to think for a second about what traits do you have that you know that you inherited? And where do you think that these traits would come from? In the picture on this slide, inherited or not, you have a list of things that are inherited that you receive from your parents, and an example of traits that are not inherited. Most are learned traits. So the ability to speak a language, the ability to read music, painting, playing an instrument, your hairstyle, all of those things that you learn as you get older and go through life, they are not inherited. The children are not born knowing those things. Gregor Mendel is considered to be the father of all genetics. He was an Austrian monk that worked in a garden studying garden peas and growing pea plants. Pea plants have very clear, distinct traits. So working with them to study genetics, it was a perfect plant for that purpose. So he chose them because they are self-pollinating, which means that they can fertilize themselves, easy to experimentally cross-pollinate. So it was easy for him to choose which flowers or which plants to breed with others. And they contained very clear traits, pure purple flowers or pure white flowers. And a trait is just any single feature of an organism. So here we have a picture of pea plants, and some of them are tall and some of them are short. In the first generation, or the pea generation that we call the parents, Mendel bred a tall plant with a short plant. And all of the babies that he got from that were tall. All of the kids from that parent of a tall parent and a short parent, all of them 100% were tall. We call that the F1 generation, the first filial, and that's the kids. When he took those two tall plants from the F1 generation and bred them together, the grandkids were, 3 of them were tall and 1 of them was short or dwarf. And so that was the F2 generation, or the second filial. So if you're paying attention, you'll see that this short trait, it skipped a generation. We saw it originally with the parents, but it didn't show up in the kids, and then it showed up again in the grandkids. We call traits that hide behind other traits recessive. And the trait that they hide behind, we call that dominant. So in this example, our tall pea plant was the dominant trait and the short pea plant was recessive. And if you look at this picture now, you'll see that we have letters underneath the description that represents what was shown on their genes. So in sexual organisms, each of them have two of each trait. They're called homologous pairs. We used the word homologous pairs when we were talking about chromosomes, especially during the meiosis unit. One of each pair is passed on to offspring by each parent. So what that means is that when you have this tall pea plant, capital T, and the short pea plant, lowercase t, one of the tall t's and one of the short t's goes to all of the offspring. So all of them are tall with one dominant trait and one recessive trait. Because it is a recessive trait, it will not show if it is combined with one of the dominant traits. The dominant trait will show. When you then took 2 of those tall offspring from the kids that both had a dominant trait and a recessive trait, you can see that the grandkids have a combination of different possible combinations of the big T, that dominant trait, and the little t, that recessive trait. And only when you see 2 of the little t's together, both recessive, you inherit the recessive trait from both parents, that is the only time you will see that recessive feature showing up, or that short trait. Different forms of the same trait are called alleles. Your alleles here would be for tall or short, different forms of the same trait. The trait that is being studied in the experiment is plant height. So the trait is plant height, and the alleles are for tall, which is dominant, or short, which is recessive. What are the 2 alleles or forms of the trait being studied? Mendel didn't just study height. He also studied flower color, flower position, seed color, seed shape, pod color, pod shape. He studied a variety of different traits, and they're using another word here in place of the word allele. The term that is being used is variance. So I want you to try this one on your own, and there is space in your notes to do. What trait is being studied? What is the dominant allele of the trait? And what is the recessive allele of the trait? Go ahead and write down all your answers before we move on. Now I also want you to put down the percentages for each generation. For the P generation, what is your percent tall and what is your percent short? For the F1 generation, what is your percent tall and what is your percent short? And then again for the F2 generation, we need to know what your percentages are for tall and short. Hopefully you received 50-50 for tall and short in the P generation, 100% to 0 in the F1 generation, and 75-25 in the F2 generation. So to explain why that happened, Mendel proposed that there are different factors, and that's what he's calling the alleles, segregate or separate from the pairs to be passed on to offspring from each parent. Mendel's hereditary factors were genes, and he didn't know this at the time because DNA had not yet been discovered, genes had not yet been discovered. We didn't know exactly how or what mechanism was used to pass things on. So today we can show exactly that genes are located on chromosomes. The location of a particular gene can be seen by tagging isolated chromosomes with a fluorescent dye that highlights the gene. Here is a picture that shows chromosomes, and specific genes have been highlighted or tagged. These little bright yellow spots right here on the chromosome is a specific gene that we are trying to isolate, and now with this fluorescent dye you can see exactly where it is, and now you know that these two chromosomes right here are the homologous pairs of each other. So I'm going to need you to think back a little bit into the meiosis unit. We were talking about how all the chromosomes separated and lined up. Mendelian genetics has its basis in the behavior of chromosomes, and how they separate was explained when we talked about meiosis and how you make your gametes or your sperm and your egg. So that was first described in the 1800s, and the chromosome theory of inheritance states that all genes have specific locations on chromosomes, and that chromosomes undergo segregation and independent assortment. Looking here, let's review the laws of segregation and independent assortment. This is a photo of cells undergoing meiosis. Remember that in prophase 1 of meiosis, they find your homologous pairs are going to pair up with each other. That means your one from mom and your one from dad find each other and line up. These are then pulled apart in anaphase 1. The homologous pairs separate and pull apart in anaphase 1, and that is a law of segregation, where you separate all those homologous pairs. Remember that in metaphase 1, you have 2 rows of 23 chromosomes, one from mom, one from dad, all lined up, and then they separate. So now you don't have that combination. You do not have those 2 alleles available anymore. You just have one over here and one over there, and you are making multiple gametes. In your law of independent assortment, that just says, okay, we're lining up mom and dad's chromosomes next to each other. But they each line up separately from each other. They have nothing to do with each other. So chromosome number 1, if mom's is here and dad's is here, that doesn't impact how chromosome number 2 is going to line up. It can be mom and dad on the same side here, or they can be switched. They all line up independently of each other during metaphase 1 or prophase 1 when they come together. And so when they separate, it's all shuffled up. Every time you go through it, you're shuffling it and randomizing your possibilities, your genetic assortment. Here is a picture of anaphase 1. This little animation of meiosis is just to remind you how it works really quick, and that homologous chromosomes split up in anaphase 1 so that only one allele of each trait gets passed on to your gametes. So the pair of alleles you have for any trait is called your genotype. We're going to give an example of nose shape. We're going to pretend, this isn't real, okay? So we're going to pretend that having a round nose is dominant to having a pointy nose. And because we are using a trait nose shape, we'll use the letter N to represent the traits. Your dominant allele is represented by an uppercase letter. So if we're using the letter N, your dominant allele should be a capital N. Your recessive allele should be a lowercase n. So your possible combinations would be your genotypes. Remember, you have two combinations. You can have both capital, one capital and one lowercase, or both lowercase. If they are both capital N, we call that homozygous dominant. Remember, the prefix homo means the same. And dominant is telling you that they're capital. They're both dominant traits. So you have the same alleles, and they're both dominant. That tells you that it's two capital letters together. The capital and the lowercase together is heterozygous. Hetero means different. So you have one dominant allele and one recessive allele, one capital letter and one lowercase letter. And then you have homozygous recessive. Again, homozygous tells you that they're both the same, and recessive tells you that they're both the recessive allele, or both lowercase. So your genotype options are, again, homozygous dominant, heterozygous, or homozygous recessive. So how did you get your genotype? You get one letter, one allele from mom and one allele from dad. So one is carried in the egg and one is carried in the sperm. Each set of alleles is a homologous pair. What would be this person's genotype? We've talked a lot about the genotype, but we need to talk about something called the phenotype. The phenotype is the expressed feature you can see. So the genotype is what's written in your genes. It's the letters that are written in your genes. The phenotype is your physical feature. What does it look like? So how is it expressed? What kind of nose do they have? We're still talking about nose traits here. If it's capital N, and round nose is dominant to pointy nose, then they have a round nose. If you have capital N, little n, what is the person's genotype? And what is their phenotype? They are heterozygous because they have two different versions of the allele. And they still have a round nose because dominant traits overshadow recessive traits. Then this person's genotype, homozygous recessive, and here's the first person with a pointy nose. Recessive alleles show only if no dominant trait is present. So Gregor Mendel did a lot of research and wrote up three principles. Principles of segregation, independent assortment, and dominance. We already discussed them a little bit, but we're going to go through them one more time. So his principle of segregation is that the parents' alleles for a trait would separate during the formation of egg and sperm. So each parent donates an allele to the offspring. This is talking about meiosis, guys. This is when this happens. Your alleles separate during meiosis, so that you only get one of each allele in each egg and one of each allele in each sperm. So when they join together, you have two. Here you have a father that is heterozygous, capital R, little r, a mother that's heterozygous, capital R, little r. So the dad's sperm are either going to have a capital R or they're going to have a little r. The mother's egg, same thing. They're either going to have a capital R or they're going to have a little r. And so you get any combination of the two, one egg and one sperm. So you see this first baby on the left was able to get the dominant trait from both parents. The second and third baby got one dominant trait from one parent and one recessive trait from another. And then the last baby got both recessive traits. And we are going to learn how to show that in a Punnett square. The principle of independent assortment means that genes for one trait do not affect the inheritance of genes for another. For example, height will not change seed color for pea plants. So this is a picture of Amy Adams. She's in movies. And she has blue eyes and red hair. So if she has a baby that has blue eyes, just like her, would her baby also have her red hair? And, and the answer is maybe, maybe not. They have nothing to do with each other. That's what independent assortment is. The allele for hair color has nothing to do with the allele for eye color. They are separate, okay? They are in separate areas and they assort independently of each other. And here is the principle of dominance, where one allele can mask or hide another allele. The dominant trait is the allele that masks or hides the effect of the other allele. And so if you have a dominant allele, then that's going to hide the recessive. And a recessive trait is the gene that can be masked by another allele. And in our tall and short pea plant examples, the tall plant was capital T, the short plant was lowercase t. I see this as a typo here on the PowerPoint slide, but just go ahead and cross that off. The short plant should be lowercase t. Okay, make sure it's right in your notes. So here's another example where we're talking about flower color. So go ahead and answer, what's the trait? What are the alleles or the versions of the trait? And which one do you think is dominant? And you have your genotypes listed there. And you also have the phenotype, which is the picture, which is what you can. So we're going to set up our first Punnett square. You only get one allele from the egg and one from the sperm. So only one can be passed from each parent. If the mother's genotype is capital lowercase, then what is her genotype? Heterozygous. If the dad has a capital and lowercase also, what is his genotype? Heterozygous. So we're going to look at the possibility or the probabilities of having different babies from the combination of the different eggs and sperm. So in this top left corner, we're talking about what if this egg, capital P, gets fertilized by this sperm, also capital P. And you would fill in, you go straight down, straight over to the right with capital P, and that would be a purple flower. So what if this egg meets this sperm? You have capital P, lowercase p. If you have one of each, the capital goes first. That's just how we do it. It doesn't matter which is, you don't always have to do the egg first or the sperm first. If you have one of each, the dominant one goes first. What if this egg meets this sperm? You again have capital P, lowercase p. And the last one, this egg meeting this sperm, you're going to have lowercase p, and that's going to be your white flower. Punnett squares are going to allow us to predict probabilities about offspring. We are going to be doing a lot of them in this unit, so you're going to want to know how to set them up. Make sure you practice. We're going to do a lot of different types, so you need to get very, very comfortable with it. You can predict the percentages for both the phenotypes and the ratios for the phenotypes and the genotypes. Keep in mind that they are different. So what percentages does this Punnett square predict? How many are going to be purple? Hopefully you have about 75%. 3 out of 4 is 75%. How many are going to be white? Hopefully you have 25%. 1 out of 4 is 25%. How many are going to be homozygous dominant? We're looking at the genotype now. That is capital P. We only have one of them, so 1 out of 4, again, 25%. How many are going to be heterozygous now? We have 1, 2 out of 4, so capital P, little p, that'll be 50%. And last but not least, how many are going to be homozygous recessive? 1 out of 4, 25%. So what is the probability that this couple will have a baby with the little a disease? So the diseased allele, the healthy allele is capital A. The diseased allele is lowercase a. And their probability of having a baby with the little a disease is only going to be about 25% or 1 out of 4. Because even if you have one big A, that is the healthy allele, that is the dominant trait, and that is what's going to show. These children that have one of each, that are heterozygous, are going to be called carriers. And a carrier is a heterozygote that can pass on the trait but doesn't actually have the disease. This year we watched Lorenzo's oil. The mother in that was a carrier for ALD, the disease that Lorenzo got. She didn't have it because she only had one diseased allele, and she still had a healthy allele. Lorenzo got it because he received that from her, and so he only had the diseased allele. So for disease A, what if a homozygous dominant man marries a carrier woman? So homozygous dominant man, what would dad's genotype be then? Hopefully you're able to put capital, capital. A carrier woman would be heterozygous, capital, lower. So you're going to set it up with dad's alleles down the side and mom's across the top. And it doesn't matter if you switch those, just separate them here. So capital, lowercase, and capital, capital. Now we're going to fill in the boxes. These should be right across from each other and not up and down. Okay, so big A right next to each other like over here. Big A, little a right next to each other like it has it over here. Again, big A, and if these two meet, it'll be big A, little a. So your genotype percentage is big A, you have 2 out of 4 that have it, so that's 50%. Heterozygous, big A, little a, 2 out of 4 have it, and they should all look like this one here. So that's 50% again. Little a, homozygous recessive, 0 out of 4 have it, so that's 0%. The offspring phenotype, healthy versus diseased, well, 100% of these offspring are healthy. 100%. Because in order to have the disease, if the disease is a recessive trait, they have to be little a, and none of them are. So to be 100% healthy and 0% diseased. Now, if you want to separate how many of the offspring are carriers, then the heterozygous offspring are carriers of the disease, and they can pass it on to their children, but they don't have it. So 2 out of 4 are heterozygous, meaning that 2 out of 4, or 50%, are carriers. So what if a carrier man marries a woman with the disease? Dad is a carrier, means he's capital A, little a, and a mom has the disease, means she's little a. So if we set that up, mom across the top, dad down the side. This first box, heterozygous, big A, little a. Second box, same, big A, little a. Third box here, 2 little a's. And fourth box, again, 2 little a's. So if we look at our genotype percentages, we're going to have 0% homozygous dominant. And when you're doing a lot of your homework stuff, it is okay with me if you abbreviate the way it's abbreviated here. Capital H, capital D for homozygous dominant, HET for heterozygous, HR for homozygous recessive. That is fine with me, but you do need to know the full term. You just don't have to write it out fully every single time. That would be crazy given the amount of punnett squares you're going to be doing. So capital A, we've got nothing. That would be 0%. Capital A, little a, or heterozygous, we have these top 2 boxes that are capital A, little a. So that's 2 out of 4, that's 50%. Homozygous recessive, 2 out of 4, which is 50%. So what percent is healthy? The 2 that are heterozygous carriers, they are both healthy. So 2 out of 4 is 50%. 2 that are diseased are these homozygous recessive ones, little a. So that's 2 out of 4. So that is going to be 50%. The percent of the healthy ones that are carriers, 100% of the healthy offspring are carriers. So it's just talking about how many of the ones that we said are healthy, so it's these 2 here, are carriers. And so we're not talking about the diseased version at all. So that would be 100%.